microorganisms to be around there. Otherwise, we just have like a pile of trash lying around us, and this food waste would just be lying around us if it weren't for the detritivores or the decomposers, which is mostly the bacteria and fungi. And so around that time, I thought, no, I think we really need to go back and tell people that yes, some microorganisms are pathogenic, but most of the microorganisms have a really positive role to play with everything we talked about, species and their role in the environment and in our life, and how they indicate a healthy environment, or how every species has a role to play in the balance, particularly in the nutrient balance, which is one of the most important principles of sustainability, but that species also being a part of biodiversity, which is naturally present in our planet. So that's when I sort of designed this lecture. And also I wanted to connect it with how these microorganisms are found almost everywhere on the planet. And there, of course, they have a role to play as well. So the spheres, and then in terms of the spheres, the nutrient recycling, they're also habitats and the importance of these microorganisms as such. And even how, for example, sometimes the phages are used, which is a virus, to create antibiotic resistance. So there are a lot of applications even in terms of fighting these pathogens where some of these microorganisms help us. So just acknowledging that role of these microorganisms, and maybe we can go back to yes.
So we can classify them as viruses, bacteria, and all of them have different roles. All of them look different components. So if we judge basically from size, then viruses are the small. And as you said, okay, most of them are like the part of them are pathogenic, most of them are pathogenic. If we're dealing with that, right? So that's so it regulates the nitrogen that is in our environment. Or if we're dealing with, let's say, viruses, then we have bacterial phage, right, which attacks the bacteria, which also equals like generating. So, and fungi, like like mushrooms. So yeah, they're like very useful if we know what to do with them. And
great. Thank you. Anybody else? Yeah.
I believe in biotechnology, microorganisms are used, for example, to produce insulin in a large level. But I think they splice the gene and insert it into the microorganism.
Yeah.
And mass produce insulin.
So, for example, whatever is super bad when it comes to parasitism, and then, so for example, for this virus to go to enter our body, find its way to a living cell, replicate there, and then, so for example, that virus eventually enters your body, one of the cells, and asks the cells to replicate its RNA for the virus. And so when it has multiple replications, the cell kind of bursts. So when we say it has attacked the host cell, essentially it's now held the host cell hostage and is asking the cell to create its multiple copies because the virus doesn't have the ability to reproduce. So the human cell is doing that job for the coronavirus. And then when the cell is bursting, you have multiple copies of the virus, which can then go and attack the cells, of course, depending on which cell it's going to. For example, in this case, it's lungs or the liver, then you can negotiate more around that as well. But in case of parasite, it's really bad. But if you think about, for example, vaccine or insulin or penicillin production, the same function of reproducing at such a fast pace is really useful because then you can create a lot of what you want. So yeah, again, like based on function, it kind of varies as well. Before we start, I think we can go back to what we studied in lecture 2. And in lecture 2, we kind of discussed what is the evolution of life. And remember, even there, I told you that viruses are so old. They have evolved, despite the fact that they live between dead and living, and just lie around. The fact that they've evolved in such a long time, kind of means that they are that they've been around and they probably are better and know the world better and have adapted better to how the world is changing. And oftentimes our research has a part to play as well, and the way we live. So all these chemicals, all these heavy metals that we expose these microorganisms to, we help them fight, we help them resist. And that's why I think a contaminated environment is at times really, really bad. If there's a bio which can be converted into a super bio, because you've now given it all the exposure to all the drugs that are around in pharmacy. But I think I wanted to just come back to generally, like when you see a division of how life is divided, you will almost always see like prokaryotes and eukaryotes. So prokaryotes is sort of like unicellular, not really established, had much older, but have a lot of classification, probably billions of species, most of which have not been discovered. But eukaryotes are more multicellular, so for example, us, the fungi, the plants. So that's generally like the word that's used to describe the more established cells and then the less established unicellular cells. Okay, so that was just that. And then if you look at it visually, so sometimes we just call these archaebacteria as well, but they have some more important functions than bacteria in itself. So for example, a lot of mutagenic bacteria come under archae, but very often they're just clubbed as bacteria. But you would see this definition in a lot of classification studies as well. And of course, within them, then there is a lot of division in each group. And like I said, the eukaryotes are more established, and then plants, animals on the exception. Okay. Right. So just to re-emphasize what's already been said, some of you can sometimes look at the screen so that I know you're not looking at the phones. So the microorganisms and whoever I'm calling that is not still looking at the screen. Anyway, so microorganisms could be divided into bacteria, protozoa. So like I said, between unicellular and microorganisms are organisms that are not visible with naked eye, but they're not always necessarily unicellular. You know, some multicellular organisms could also be not visible to the naked eye. So, and generally, like if you go really, really deep into classification, some algae are not visible. It's only when they join together, like join hands together, that you actually see them, but they're still classified into in the microorganisms. And similarly, a lot of fungi, of course, the spores are not, so there are a lot of spores around us right now, fungi, but we cannot see it. But there are also a lot of funguses that you see around, which is visible. So they're not micro, they're macro, and so on. And similarly, some parasites. So because they generally, when their eggs are not visible and they're really smaller size, they're now being categorized into microorganisms. And as we discussed there, and I didn't go into that, and I don't know why I didn't go into that, but in all of your elementary science, you were taught what's the difference between a human cell and an cell. What is the difference between, for example, the viruses, the bacteria, and fungi? So those cells have evolved a little bit. So if you look at the viruses, you say it doesn't have an evolved cell. It basically doesn't have anything to do on its own. It's only able to produce particular antigens, like we said, and it has a DNA or RNA, but not necessarily like established. Generally, it's just covered by a layer, a protein layer, and sometimes it has a lipid layer as well. And you can play, like, it can give you a lot of information. So for example, if you think of COVID, just having that lipid layer means that just washing your hands with soap will protect you, because this particular virus now has a lipid layer around it. So it's important to know what the characteristics of each of these microorganisms are, because that's where then you can control or manipulate it for your own use in life. Yeah, so, and I think same for bacteria, because I think you've studied bacteria a lot. Funguses are more evolved, they have a proper nucleus. Bacteria do not have a proper nucleus, they do have a cell wall. but their composition is also very. And then as you move to a human cell, there's a cytoplasm. There are a lot more organelles. Those organelles are not as established, for example, as compared to an animal or plant cell. And then those cells have differences as well. But all of those things I haven't really outlined because I thought if you do not remember it until now, there's no point in taking that to you again. But it'd be nice for you to just go and quickly understand what the differences were. Yes, there was. I think with washing, it's the cover and that attachment is more better when it hits, like when it is entered your cell wall. The washing part is applicated when it still hasn't entered the body. So if it is outside on the skin, I think it's difficult for it to attach because it's still like between partly alive, partly dead. The moment it leaves a living body, it's gone back to the line of partly dead, partly alive. So it will only become way more powerful when you let it in the body. And you guys then give it access to the things that I don't think I haven't seen it because skin also has objective where they wouldn't allow the virus to just. So us point here we can use that, we can manipulate that just because of the different function of virus. Because I think this is something that also really bothers me, but every research that I've seen says that these are like the sources that are viruses, and that triggers that. It's like a half, I don't know. Okay. So basically, all of these cells have different, like I said, they'll have different compositions, different morphologies. Just like Behram mentioned, they have these spikes now, which make them even more powerful than some other viruses. Bacteria, some of the archaea have these flagella that help them move around. So all of these characteristics and means of movement, and then the reproduction as well, of course. So for example, for fungi, we say that the spores that are formed can also lie around for as long as they like, because they form this protective dormant layer. And let's say it's just lying around the room, but the moment it finds a bread, the moment it finds a food, it just go and try and replicate. And that will, I think, for example, in fungi, there is budding. In bacteria, there's asexual reproduction. So a lot of these characteristics then differentiate them from each other, and that's why we've categorized them into different categories. Otherwise, we could just call them microorganisms. So they have both. Yeah. I think because still they're different from one another, different in a sense that no matter what is happening, whether it's, for example, just the categorization of fungi into multicellular is different. But I think most of the microorganisms, most of the micro fungi, they generally use the mechanism of fungi. Most of them. I'm not saying all of them. And similarly for bacteria, yes, like you could say between mitosis and meiosis, but I think most of it, again, I think the dominating side is asexual, and there are no clear definitions. But again, if you go back, and maybe that's why there's no single mechanism for identifying them from one another, because I'm not saying that reproduction is the sole criteria for dividing them into groups. It's also how they move. It's also their morphology. But then I think the major thing is their cell compositions. Their cells are entirely different from one another, and the cell compositions group them in one, and group these in the other. and of course, then how they live, and you know, then it comes back to why their habitats are so diverse. That you know, we can't even imagine. Even though human beings have also become super powerful, but I think microorganisms still win because of how old they are and their ability to reproduce that in the environment. And maybe all of these definitions will evolve. So I think in science there is no, oh no, you're not right, because maybe suddenly we discover a fungi or a bacteria that defies all of this. So, you know, even I wouldn't be very strict with what's in the books, because a lot of these species are still discovered and maybe they will bring traits, composition, and in the way that they do. So as science evolves, a lot of these characteristics can change, but because in such a long time it hasn't, we sort of now categorize them according to what literature and scholarship suggests. Okay, so I think some of the traits that microorganisms have that we keep coming back to the size, it was also mentioned earlier in class. We also keep talking about diverse habitats. So I think we will talk about the size and why it matters, and why it matters in terms of controlling these microorganisms. So yesterday, I think it was Danya who mentioned that maybe in the pilot, I don't know, not in the chat, definitely in the pilot, about the size of viruses and what kind of masks would be suitable to stop these viruses from entering our body. Now, I have been using cloth masks for as long as I remember, since COVID. So in the first month, only I used surgical masks, and past that, I didn't see any use if you wanted to use it. So I switched to cloth masks, and I've been using that masks ever since. It has been really effective because I think a lot of the calls for this, where you go while you're there, and, you know, staying away from crowds and all the So I've been, you know, really protected, I think, because I also observe all the other SOPs. But why do you think would I claim that micro mask is as effective as a surgical mask? And the answer is partly here, but also it's not partly here. We will come back to it. Yes, I have. And why do you think that? How so? Because the virus does not have the ability to fly. So how does it reach? Yeah, so the droplets. So for example, if I'm standing next to you and I'm breathing, then it's probably like the vapors that are coming to you. And you need to stop just those. And most of those vapors wouldn't be the size of a virus. They wouldn't be, for example, this size. They're probably much bigger in size. And for that, you just need a barrier that will stop the droplets. And for droplets, I personally think that clothes are way more effective than any other kind of mask. Because if you have a mask that doesn't absorb moisture, then it could be the finest mask around, but it may not be effective at stopping the virus. So a lot of biologists and microbiologists actually keep saying that your cloth mask is enough. Of course, you need to wash it. Of course, you need to make sure that you're not just wearing the same mask. It's not stuffed enough to just get the moisture to come to you. So you'll have to do all of those things. But I personally think, and a lot of microbiologists also think, that it's okay if your mask has the ability to absorb moisture, to absorb the vapors, and not allow it to come through. Otherwise, if you think about size, so your masks probably are in the micro range, right? So somewhere here. And if you look at the size, viruses are much smaller. They're in the nanometer size. So just to bring it back to maybe filtration, if we define it in terms of how can you stop this particular size, let's say in water filtration or in air filtration, the size of filter that you would use would go from microfiltration to ultrafiltration to nanofiltration to reverse osmosis. And in microfiltration, we say this is the size that it would stop. If you want to stop, let's say, the enterovirus, you will have to go in the ultrafiltration. If you want to stop, for example, just the bacteria, microfiltration should be enough. But this is in terms of, for example, forcing a water molecule out, allowing the air particles to pass through the filter where you're forcing it to take out. For us, it's, with the mask, it's not as controlled. You're not, the inlet and outlet, they're not a box. A lot of it, for example, a lot of the air would just go from the sides directly to our nose. We don't really know. And of course, we can discuss what kind of mask should we want to make sure that only the air that you breathe through the mask goes into your lungs. But I think the size is important because when we come to the control of pollution, and not just microbiological pollution, but all kinds of pollution, you need to have a sense of scale. And that sense of scale is often combined with the size of the filter. So how fine, so if it's a fabric, it will be super loose, but it will still allow, for example, for sand particles to be on the fabric. Or for example, for your boiled water, you still see a lot of the calcium, magnesium compounds kind of just settling on the floor and not going down. So their size is probably way higher. But here, if you come back to the scale and look back at what is it that you want to stop and what kind of filtration would you need for that, I think it's that size comparison is really important. So nanofiltration then, it just brings you, it should only allow the particular atom to pass through. So for example, in reverse osmosis, which is one of the filtration techniques, where the membrane is in the nano range, we say that ideally it should only allow water molecules to pass through and nothing else, like absolutely nothing else. And sometimes, and we had this discussion in the last class, where once you have water molecule passing through RO, it's demineralized. Everything that was a part of it is removed. So now anything that's coming out will have to be remineralized. Because your water molecules aren't just H2O, they also bring you a lot of other important micronutrients, which are part of the water, the natural water that comes in the streams. And maybe that's why if you look at your water bottles, so maybe Roja can help us with what are some of the other ingredients that the water bottle says, and maybe also like the quantity of a particular component. And basically what they're telling you is once they filter it and got rid of all the extra material that is there, here are the minerals that they added just to make sure that your body and its needs are met.
Chloride 10, 100, sulfate 20,
Right. So they're telling you that we also maintain the pH, we are adding sodium, magnesium, all of those minerals which are required in certain quantities. It's not just the H2O that you need, but also kind of mineralize it. So I think with the filtration techniques, it's important, for example, for the air, you probably just want clean air, but what is it stopping? So it would also be nice to go back and look at what kind of filter. So when you say that a particular, so for example, I know that a lot of you are doing articles on CO2 removal using machines. Some of you are looking at different technological interventions. It would be nice to go and see that what kind of filter is used in that machine. Is it just a filtration technique, or are there different, or is it a cation exchange method, just to go a little deeper into text, where different ions are used for a particular chemical to be absorbed or rest of them to be released. So, like, what is the mechanism that's going in there? And again, it would bring me back to the assignment that if they tell you that this particular machine is now making the air clean, like how clean? How do they define clean? What is the composition of the air that's going in, and what is the composition of the air that's coming out? And is that kind of information given to the reader? What is the filter made of? Where is it coming from? Where will the filter be discarded? So the biggest problem, because I also worked on wastewater treatment using membrane filtration, the biggest criticism that we face, for example, in reverse osmosis, is the membrane is going to eventually chop. There's a lot of energy that needs to go into high pressure for the water molecules to pass through. So first is that. Second is that eventually these membranes are going to chop, which means that you need to keep replacing them. So a lot of people criticize that it's not a sustainable solution because sometimes when the suspended and dissolved solids are really high and you're applying a lot of pressure and you're not trying to maintain the membrane, could go, it could clog in the first week of its operation. So how do you then use a sustainable scheme? And of course, there are ways about it which we can discuss later as well. But just I think the size in itself, particularly with reference to any discussion of microorganisms, is really important because it then gives you an understanding. And maybe I can take a break and take any questions, comments, and yes. Yeah, so for the virus to enter your body, it's important that the water vapor kind of enters your body. The water vapor that contains, or even the dust particle that the virus is lying on, should get into your body. And if it manages to get into your body, you're infected. I'm not saying that cloth masks are 100% effective. All the masks are not 100% effective because I personally think that a lot of us, for example, I tend to do this all the time. When the mask, probably this side of the mask is more exposed to the virus or bacteria. If I keep doing this, like already my hand has some of that moisture, which is why like you're stopped continuously from touching your eyes. And so I don't think any of them are 100% effective. They're just the means to delay your exposure. Eventually, for example, if you are sitting in a stuffy room with a lot of people, oops, that could be our class, then for example, this side where the air is simply just coming in, if the moisture content is really, really high, if it's super humid in general, then automatically you will be eventually exposed. So, and I think there were a lot of interesting studies on how many people should sit in light when COVID happened. I read this really nice article where they had modeled. So if you're sitting in one corner of the room and I'm sitting in this corner of the room, and we are sitting there for 7 to 8 hours, then given the size of the room, if you're infected and I'm not, what is the time that it will take for the air to be humid enough for the virus to eventually reach? So there are those interesting studies as well. Yes, Asa.
My understanding criteria is like one clear factor that make the most budget is the produce at a very fast rate. But जो आपकी नई bacteria होती है उसके like they can become like resistant to like a lot of antibiotics, for example. But are there like other factors or other characteristics of bacteria which allow so abundant or like the rate of production?
Yeah, I think all of those characteristics that we're generally talking about of their ability, their size, first of all, their ability to travel, their ability to adapt to different environments, their ability to, for example, adapt to drugs. So with reference to our sphere, the anthroposphere, I think it's their ability to reproduce fast and acclimatize to any environmental condition that we expose them to, including drugs, is, I think, a more powerful trait. But that trait then comes from the ability to, for example, cling onto surfaces, to form spores, to be lying dormant around at a certain time, to wait for the right time, finding those spaces. I think, again, it's very similar to the answer that I gave before, that there is a lot of those characteristics, the ability to move around environments, which could add on to that. So it's not just the reproduction, because of course, all of them have the ability to do. So what makes bacteria more powerful is their size and the diversity of species, and then the diversity of the habitats in which they survive. Yes.
And
all species have lived on in this planet and all of that the habitats have changed.
Yeah.
Can we say that microorganisms are some species that have that they don't have a specific habitat because they can survive in any habitat. So even all other 90% habitat systems microorganisms survive together. So are not they more powerful than humans in terms of survival and all other species?
They are and they're not. So I think that's why if you read the book, or I've also from time to time asked you, how have they become such powerful species? We have created that system, and you can define it in terms of our rates and ability to think and create system. But they also live in populations, and that ability to live in populations and fight allows them to stay in it. So I think because they aren't as evolved as organisms as we are, it probably, for example, they not have a brain, although they have all of these abilities to sense, to move, to understand, giving these. So in bacteria, there's this wonderful, I love the concept. It's called quorum sensing and quorum quenching. The bacteria talk to each other. They sense chemical signals. They sense threats exactly how, for example, a bigger, more evolved species in the oceans would do. They eat themselves sometimes, So just like the ability to stay dormant in a certain time, they also have the ability to destroy themselves at times. So I think all of those concepts also are really, really beautiful as well. But I wouldn't agree to they can, not all the bacteria survive everywhere. I think it's important because I wanted to create something like this myself, but because I found it on the internet, so I was happy that it saved time. But when we were discussing in the second lecture, someone asked about the diverse habitats that we have. And this graph, this picture essentially shows that they are super diverse, but it doesn't mean that the acidophiles, because they have a particular pH up to which they live, I think in a more alkaline condition, they wouldn't be able to survive. So it's not true that the acidophiles will survive in alkaline conditions, or those that have the ability to survive in, for example, cooler temperatures or warmer temperatures, and they have different names. Maybe the names are not that important, or maybe they are. It's up to you to want to remember them. But what's important is that they have. So, for example, its optimum pH is 8.5, whereas for an acid mine drain, which is coming from the mines, where because of its acidic nature, it accumulates a lot of, it leaches a lot of those heavy metals, gives a color. So some of you might have seen outside of, or the rivers that flow around the mines, you would have seen these colored rivers. They're essentially the acidic nature of the solution, and that solution being able to wash or leach a lot of the chemicals away from it. Highly toxic. I think that's why some of the mining activities are really, really criticized. But the bacteria that would survive there it's an acidophile, so it needs acidic conditions. Would this same bacteria survive here, where the pH condition is 8.5? I don't think so. Some might, not all. It depends on the kind of bacteria. And then for some, there is a temperature optimum. So these cycrophiles, they only survive in temperatures less than 50 degrees. So it means that they wouldn't be able to survive at temperatures greater than 45. So it's true that they have habitated diverse habitats, but I think they have limitations. So that is their environment. That is the environment where they will thrive the best. And it could be, and all of these are generally categorized into extremophiles, which means that they love extreme conditions. But it doesn't mean that the bacteria that we find around us in abundance are not. Of course, they play part. A lot of these bacteria help degrade some of these chemicals. So they have really important parts wherever they are. But in abundant, nutrient-rich environment, for example, in a soil, we say that a spoon of soil contains billions of bacteria. It also has a really important role as well. So, yes, there are a lot of extremophiles, and yes, they're important Again, yes, bacteria vary in size and in the kind of habitats that they live in, but the overall bacterial population around the globe is also important. So it's not just about extremophiles, it's about all kinds. Maybe it's related a little to what Asad was talking about, of how they have become powerful. So I think this is then another discussion, and maybe any of you who's read about it, we kept talking about methane, we kept talking about how animals, or maybe we didn't, I don't remember, maybe we did talk about how animal farming or industrial farming or industrial food production contributes to emission of methane. And that is actually related to, for example, here having those bacteria, those methanogenic bacteria that release methane. And I think this kind of says it all of how between the food and whatever is happening in the stomach, you have these bacteria which are anaerobic in nature. Anaerobic means they can operate without oxygen, then converting all of these nutrients into and it's called methane-belching bacteria, so they release methane. But the same, if you remember, we also talked in the biofuel and biogas slide, we talked about how the cow dung is actually used in methane production. And it's because some of these methanogenic bacteria are already there. We just need to give them the perfect conditions, which they get here in the anaerobic digestion of the biogas, where we allow these methanogenic bacteria to then release. So in a way, it's not just negative, it would also be positive. And we were talking about habitats, so I thought it would be important to also talk about them not only being in the environment, in the different spheres of the environment, but also in the, for example, within a biodiversity, within life, and how they benefit each other. So this is, I think, was it Abdullah? You discussed something like this, where, you know, in the stomach of a lot of species, there is bacteria where the species allow for the wands to be a little weaker. It prepares, for example, the plastic for the bacteria, and when it brings it to these bacteria then have the ability to degrade polyethylene. So this particular bacteria, so essentially their relationship is such that the insect would give bacteria the nutrients, water, etc., and then the bacteria would give something back to the insect. So it would give, for example, vitamin K to, depending on the species, it would give a certain nutrient back to its parent organism. But the same relationship is manipulated in wax worms or some of the other worms, where they have the tendency, or researchers prove that they have the ability to degrade because of the kind of relationship that they have. And the bacteria are bacillus species or Pseudomonas species, and a lot of these species that could survive in the stomach, or let's say inside of the wax worm, or some of the enterobacterias have the ability to then degrade. We'll talk about the rates of degradation and then how effective it is later. But here again is someone just cupping and a droplet analyzed. So within that one droplet, teeny tiny droplet, that's like the number that they can get. Beram, can you give me some time? So I'll try and finish this and maybe come back to your comments, because I think I have very little time. Yeah, so I'll come back to some of the questions and comments once we finish this. So we talked about size, we talked about habitats. Now we have to talk about relationships. And I like this topic because you can also apply it in your own life, and some of the relationships that you have in your life. And I like that exercise of finding who the parasite in your life is. Like, don't do that. But it'd be nice to see like what kind of relationships you have. And bacteria and microorganisms generally have these really nice relationships, where sometimes one benefits the others, where sometimes both benefit each other, where sometimes one benefits and the other is just unaffected, and another time one benefits but also destroys the person that it benefits. So it's nice to just think of what kind of relationships these microorganisms have, and it's true for even for the more evolved organisms as well. So how, for example, trees, and I think I've written it here as well, trees give us oxygen, which is important for our survival, a lot of phytoplanktons. So recently you may have read a lot of articles about how trees are not the major producer of oxygen. It's actually the planktons in the ocean that produce more oxygen for life. And I don't think we emphasize that much on their importance in our lives. That kind of symbiotic relationship is really important as well. So I'll just quickly, because I don't know how much have I put in here, because generally when I create the slides, I'm super excited and I want to learn everything. And then when I'm teaching, it's too much. So this again, and try to keep thinking about these relationships. It's important that wherever I'm teaching you, you zoom out at certain points and look at the how each sphere is interacting with each other, specifically with reference to bacteria. For example, the bacteria or some of these microorganisms that habitate that are in plants, plant roots, or the root zones are now also part of the soil. So they're in the biosphere, they're also in the lithosphere. They're allowing, and if you go back, I would really encourage you to go back and read in the books the biogeochemical cycles, because some of these bacteria, for example, nitrosomas, nitrobacter, pseudomonas, they have important parts to play in those cycles and in that cycling. And it would be nice for you to go back and see, for example, how the organic matter is decomposed by these microorganisms, how that then becomes a part of the nutrient pool, how that is also required for microorganism survival, but then microorganisms kind of make it available for the plant, which can then die again and come back. So those kinds of cycles, I think, would be really important for you to think of, because I might ask these kind of questions in the mid as well. Yeah, so back to the diversity of habitats. Starting with soil, and we can just focus, for example, on fungi and bacteria. So the obvious example that you cannot forget is nitrogen fixing bacteria. And generally, when you look at leguminous plants in the root, so these plants generally have these bubbles, I don't know if you're able to see them, which is basically found in the roots of leguminous plants where there are those nitrogen fixing bacteria. Those bacteria have an important role to play in the survival of the plant, but they also have a really important role to play in the nutrient cycling within it. So fixing it from the air to making it available to the plant, which was otherwise not available, to then create the food, which is also then important for us and also important for organic matter. So all of those things are really important. But there's also, I think in terms of soil, I find fungi really fascinating, because they form these networks. So just like when I talk about bacteria talking to each other, I think the soil and the plants are really connected to each other because of the fungi. And for example, the mycelium that then forms these finer threads where a root of one plant is also connected to the root of the other. So because those particular fungi cannot create their own food, they take food from the plant, but they also make a lot of other nutrients available. Sometimes they connect different plants within a farm to each other and allow for diversity. We will, in the discussion about food security, we will look at why those tunnels, why those veins are important. Just like in our body, that those connections are important. In the soil, how then, and I think I have one image. But I do want to also mention that if the environment is changed, if the temperature of the soil is changed, if the chemical composition is changed, it could impact the population of these organisms, which could then eventually destroy the quality of the soil or the food that we are able to get. Back to this, and I don't think I need to focus a lot on this. We have talked, for example, about how different kinds of microorganisms are found in the different zones of just one aquatic environment. So in this case, for example, it's a small water body, a lake somewhere where, depending on how much light goes in, depending on what kind of nutrients are formed, in the benthic zone, for example, there could be mutagenic bacteria, but here you will have more bacteria that survive with good light and good oxygen. All of them again have gone in survival of that water body as well. So I think this you already know, but enough of positive discussions. Let's also look at how that then relates to contamination. Their abilities to live in diverse habitats, how that impacts. So air, we say, is a major way for them to transmit. Water could be a way for them to transmit as well. So currently in the discussion, in the project that I'm doing for UNITC, wherever we go, whichever farmer we talk to, they keep talking about aflatoxin. I'm not sure if you've heard about it. It's basically a fungi that's like a disease invasion crops that are found in Pakistan. And literally every farmer you go to, even the government departments, aflatoxin seems to be like the biggest problem in the food production sector in Pakistan. And you know that they spread because we've given them the ability to spread. So maybe they spread with the disease, the seeds. So if the seed has a spore of these fungi, then a seed that is transported from Punjab to Sindh could also take that aflatoxin with it. And similarly, just the air moving around and how that then impacts our overall security. But just like an emphasis on the fact that, yes, they have a lot of positive role to play, but again, like if you think about it, this is important for degradation, right? So if this doesn't happen, if we could just survive there, the field would just be lying around. From a degradation point of view, I think their role is really, really important as well. And maybe I can allow some of you to speak about what do you think are some positive roles of microorganisms in the environment. Yes, Karan. Exactly. And do we eat bacteria? Do we eat microorganisms? We have examples of that. Yes. Exactly. Yogurt is everyone's favorite. Cheese also is. There are microorganisms. Of course, like those fungus may, even yeast. After, of course, you eat a fungus on top of the pizza, but in order to make that fine dough, you need yeast, which is microorganisms. So we do eat microorganisms. And we just need to be aware of that. We just need to be made aware of the fact that this is how our lives have been all this time. But when it comes to discussion on microorganisms and science, we make it sound like we have no relationship with them or there are others where they've been a part of our body all this while. Yes, exactly. Yes, absolutely. So degradation is really important. I think it would come. I think the most important role in the environment is decomposition, or their roles as decomposers. Because without them, I don't know what our environment is. Without them, I don't know if we'll ever have that nutrient cycle, which is important for sustaining this planet. So yeah, all of that just revives, producing oxygen, providing nutrients, maintaining human health, but also, some pathogens and some pathogenic. So yeah, we kind of discussed this. Oops, this is a terrible slide, but this is what I was referring to. So you have a fungi, which is kind of connecting these, and although they're different species, but how it allows transport of carbon between plants as well. And I think this is really important. Like, I don't think when we look at soil, we imagine the role of fungi that much. We do talk about plants a lot. We do talk about the root zone a lot. We do talk about bacteria in the root zone a lot. But we do not talk about the role of fungi in these mycorrhizal associations, which I think are the nerves or the nervous system of the soil. I don't think we talk enough about it. And of course, it is a symbiotic relationship, and that needs to be highlighted. Something else that I really admire is the relationship between algae and fungi, which you see on soils all the time, and gives you these wonderful colors ranging from red to yellow, I think, and green sometimes. So basically, it's the same thing. The fungus is absorbing air and nutrients from the air, and algae is producing food, so both kind of benefit from each other. But fungi is also protecting algae from the harsh environment. So I think even that kind of relationship is really important. But like I've highlighted, the role of fungi in decomposition is really important. A quick, and I do not want you to get worried about these terms, but for example, just like they're important in decomposition of waste, the same application also applies to wastewater treatment. In wastewater treatment, without these bacteria, you will not be able to degrade the complex chemicals back to carbon dioxide. So I think that is really important. So just to categorize a few, and I know methanogenic bacteria are highlight, but for example, denitrifying bacteria, Pseudomonas aeruginosa converts back the nitrates to nitrate, the nitrites to nitrates, and then releases it back to the environment. So kind of helping in that nutrient cycle. For phosphorus reduction, for sulfur reduction, for again degradation of some of these hydrocarbons, there are bacteria available there. So for example, Nitrosomonas nitrobacter, they are nitrifying bacteria, whereas Pseudomonas is a denitrifying bacteria. So just like remembering a few of these names, but not necessarily like, oh, I don't know. But let's quickly come to virus, and let's also see how far we are, because I want you to watch that video. Okay. So like I said, for everyone else, there are cells, there are defined walls, there are defined organelles that are there, functions. Virus is a little free, even though it's super old, and maybe that is its strength, I don't know. But it is asimilar. There's no particular cell, it just has like a protein there and some DNA or some RNA. So it has nucleic acid, which is important for the survival of life. But like I said, it doesn't have the ability to reproduce, so it has to find a host cell to reproduce. But that nucleic acid is something that defines it. And for example, for COVID, we say it's the RNA virus, right? It means that it has a RNA in terms of chromosomes and adaptation. So in our cells, we have a more defined chromosome, and that double helix structure where the genetic information passes on, and then they would just have a nucleic acid, which could be DNA, could be RNA, and then it depends on the type of virus. So not for you to worry too much, but like I said, the most important thing is how they incorporate themselves into the how they find the host, how they get to the host body, and sometimes how do some of, for example, some of the flies help them reach the body of the host. So it's not always just the water droplets. There are a lot of other things as well. Yeah, so for example, I was really intimidated by this. So this research on, as you know, COVID's like a variety, evolved variety of SARS and MERS. So it's from the same family, right? And this research is way older than COVID. So I think a lot of the research evolved because some research on SARS and MERS were already happening. And if you remember SARS, and how it was associated with some of the life, and MERS was a respiratory syndrome which was associated in the Middle East with the camels that are there. And I think we can end on this note, their role in the industry. I think more than their role in the environment, which we do not appreciate that much, we do appreciate their role in the industries. We do appreciate, some of the I don't know, if you go to the Europe and you get to try some of the best cheese in the world, I think I will never enjoy a pasta in Pakistan because the parmesan doesn't taste the same. And I think you just need to be able to appreciate those kind of varieties that come from these lactobacillus that have the ability to use lactic acid, for example. But like Behram said, it's the enzymes that are more important. So even in the degradation of plastic, it's the enzymes that eventually then degrade whatever the component is that you want to degrade or convert. So like I said, yeast is really important, and they generally are categorized into Saccharomyces category. And I TA'd for a course there where the students from Chem-Eng had to create beer from ethanol. So some of these interesting applications where it could go off, right? If you do not find the right combinations. So, for example, particular groups made those beer in class, and then at the end, like some tasted better. Of course, I can taste, but for some it was apparently really good, and they took it for their friends, and for others it was just awful. So I think it depends on that as well. So if you have the right conditions, the right type of microorganism, then the process of fermentation will happen really well. And for example, in the case of wine, they do say all the time that the more it ages, the better it tastes. So I think all of that has to do with what is happening with the aging, how the transformation is taking place, and these are very important industries. They do make a lot of money, and a lot of our life depends on it. I just wanted to expand a little more on this. So cyanobacterium and their role in for example, degradation. Aspergillus has a lot of applications in industry, and then of course you all know lactobacillus. It's just for you to read later. Okay, so quickly. Bioplastic, because we talked about plastic, and I can leave the section about microplastic, which is my own work, for discussion later. But I think bioplastic is important. Like I said, it could be bio-based plastic, and that bio-based plastic is then related to biodegradation. So, for example, polylactic acid-based plastic, or even in the last class we talked about natural plastics. Naturally, there is polyethylene. Naturally, there are cellulose nitrate or other products that are created. So all of them were available naturally. They only boomed in the industry when they were created synthetically and had the ability to be mass produced and mass transported. But a lot of these plastics that are driven from natural sources, they have the ability to be degraded better, easily by microorganisms. And this kind of chemistry is called green chemistry. And maybe I can stop at the next slide and we can quickly watch the video, which is important. We kind of talked about why these bacteria are important and the role that they play. So these bacteria do allow for degradation to happen. And again, we have talked about it. I will not talk about microplastic. I'll quickly play the video. And I think